All right. Welcome, everyone. Welcome. All right. So this is actually for the recording. It's not projecting my voice, but can people hear me in the back okay? Everyone hear me okay? You can't hear me okay. I'm not speaking loud enough. This is not projecting. I will just speak loudly and then right here then. Better? Okay. So hi, everyone. I'm delighted today to introduce Dr. Suzanne Segerstrom, who is the director and Center for the Center for Healthy Aging Research and the JoAnne Leonard Petersen Endowed Chair for Gerontology and Family Studies at Oregon State University. So Dr. Segerstrom received her PhD in clinical psychology with minors in health psychology and measurement in psychometrics at UCLA. And then she also received her MPH in biostatistics from the University of Kentucky. So Dr. Segerstrom actually completed her clinical internship here at UBC in the Department of Psychology and at the Vancouver Hospital and Health Sciences Center. So welcome back to Vancouver. So prior to joining Oregon State, Dr. Segerstrom was a professor of psychology at the University of Kentucky, where she is now a professor emerita. She is also the editor-in-chief of Biopsychosocial Science and Medicine, formerly Psychosomatic Medicine. And I'm sure it was lots of fun to edit a journal while it underwent a name change for both the journal and its larger society. And so speaking of that society, she was also the president of the now Society for Biopsychosocial Science and Medicine, formerly the American Psychosomatic Society. She has received many awards for her research, her teaching, and her mentoring. And she's also been named a fellow of at least 6 professional organizations. Her work has been funded continuously by the NIH, including 2 large studies on the health consequences of motivation and goal pursuit in older women, and through a longitudinal burst design, as well as a longitudinal study of self-regulation and immunological and brain health in older adults. So I'm very much looking forward to hearing more about your research and presenting on aging, immunity, and cognition. And so please join me in welcoming Dr. Suzanne Segerstrom.
Thank you. It's great to be back here. In fact, a couple of my clinical supervisors from when I was on internship are actually here. So welcome to you guys. It's great to be back. I am trained in, and a lot of my research is in the field of psychoneuroimmunology, which is conventionally the relationship between the mind, the brain, and the immune system. And today I'm actually going to expand on that a little bit and talk about how the immune system influences and is influenced by infectious agents like viruses, and what implications that might have for the aging brain. So all of this I'm going to talk about in the context of aging. There are some seats over here on this side too. If you're looking for seats, there's some seats over here. So the context of aging. So as we age, our brains change, our immune systems change. We're more likely to be infected by viruses and other agents. And notice that in this graph, these are lines and not arrows, because these are not one-directional relationships. The brain affects the immune system, but the immune system also affects the brain. The immune system can act on viruses, but viruses can also influence the aging of the immune system. So these are intersectional. And in some of the work that I'm going to talk about today, I'm going to talk about how personality intersects with these relationships. So I'm trained as a clinical psychologist. I'm really a health psychologist. A lot of my work is in personality. Some people think I'm a social psychologist. But today I'm going to be talking about the intersection with personality. So first, just a couple of words about how the immune system changes with age. A number of different things happen. One of the things that happens is that B cells in the immune system produce less antibody when they're stimulated. And this is an important development because antibody, I almost feel like after COVID, I don't have to explain antibodies anymore. But you know that antibodies can bind to viruses, keep them from entering cells. They can also target parasites, for example, or bacteria for killing by other cells in a process called opsonization, which I just say because that's kind of a fun word to say, opsonization. And so these are data from early in the COVID pandemic. This is, excuse me, 176 adults in long-term care. One group was aged 20 to 60, and the other group was aged 80 to 100. And this is how much antibody they had after their first vaccination. This is what they were generally considering protective amounts of antibody. And so you can see that in the younger group, most people had protective amounts of antibody after the first vaccination, but a lot, in fact, about half of the older group did not. After the second vaccination, everybody in the younger group had great levels of antibody, and most, but still not all, of the older people did. So you can see these are the first and second vaccinations in the younger people, first and second vaccinations in the older people, how much less antibody they produce, how much more they rely on that second vaccination to be protected from COVID. And this is why I don't know what the recommendations are here, but in the States, if you're over 60, I believe, you can get a spring COVID booster as well as a fall booster. And this is exactly why that's that recommendation. Another thing that happens as we get older is our levels of inflammation increase. Inflammation is an absolutely critical element of immune function when it's local and time limited. And in fact, if you didn't have inflammation, say if you have a scratch or a cut, you would not get wound healing if there were no inflammation. But as we get older, we get this kind of long-term, low-grade, smoldering inflammation that's systemic. It's all through the body. And that kind of inflammation, which is produced by immune proteins called interleukins, is associated with increased morbidity and mortality from a number of different age-related diseases, including pneumonia. Atherosclerosis is an inflammatory disease. Some kinds of cancer are promoted by inflammation. Frailty is increased by inflammation. Osteoporosis. And I'm going to come back to this more later. So remember, inflammation also increases the risk for Alzheimer's disease. What you see here are some killer cells attaching to and causing the death of a target cell. This kind of cytotoxic effect is carried out by these killer cells. Or maybe I'll play that again for you so you can see it. Play it again. No? Does it not want to play again? Oh well, so you just have to look at the dead cell. So these kinds of killer cells with age become senescent or elderly. And when they do, they lose some function. In fact, most of them can still do this. They still have cytotoxic effects. But what they can't do is to create the kind of army of clones that is needed in an infection where there's not just a couple of target cells. There are many, many, many target cells. And so you need many, many, many of this kind of killer cell to effectively fight off the infection. And so I've borrowed a slide from Rita Efros, who's an immunologist friend, to really show you the importance of this phenomenon. This is her slide. Massive proliferation required. And this kind of immunosenescence can also be associated with mortality. So in this graph, people who are not cognitively impaired are the two lines on the top. This is their survival percentage over time. People with cognitive impairment are these two lines on the bottom. You can see that for the people without cognitive impairment, you have increased mortality for people who are immunosenescent. You see that difference even more graphically, more strongly in vulnerable people who are immunosenescent, the people who have cognitive problems. Okay, so why do immune cells age? Well, age is one reason. These are data from my own study. Each of these dots is a person. The people in this quadrant are not immunosenescent. The people in this upper left quadrant are immunosenescent in one kind of killer cell, a T cell. Over here, immunosenescent in another kind of killer cell, the NK cell, natural killer cell. And up here, all these people are senescent in both kinds of cells. And their colors are how old they are. So we know that immunosenescence increases with age, but when you look at this graph, it's not like it jumps out and hits you in the face, right? It's kind of hard to tell if there's a relationship with age or not. Now, that's not true in this right-hand graph. It's the same sorting, like you can see the dots are in the same places, but now you have blue triangles and green dots. And given a choice, you would much rather be a blue triangle than a green dot. It's very, very clear. And the difference between them is that people with green dots have been infected by this virus called cytomegalovirus. How many people have heard of CMV cytomegalovirus before? A few. Okay, good. So cytomegalovirus can infect the brain, the eye, the lung, the stomach, the intestines, and the kidney. And this figure says, oh, it can cause encephalitis, it can cause retinitis, it can cause pneumonia. This is true in people who are immunosuppressed. If you are not immunosuppressed and you're infected with CMV, you probably didn't even notice. Or you thought maybe you had a little bit of a cold. For most people, CMV infection is asymptomatic. And it's also a latent virus, which means that once you have it, you always have it. It's never completely cleared from your body. And that means that the prevalence of CMV infection only goes up with age. So by the time people are over 80, a very high proportion of them have been infected with CMV. So this is men and women who have CMV with age, men and women who do not have CMV, the amount of immunosenescence that they have. So you can see immunosenescence increasing with age. Ignore the 86 plus. These error bars get so big that you can hardly tell anything anymore. So you can see some increase in immunosenescence with age, but you can see much more increase in the people who have CMV. So age does cause immunosenescence, but CMV really accelerates this process. Okay, having now given you the background for all of the immunological stuff, I'm going to tell you a little bit about the thought, stress, and immunity study, which I'm going to refer to as TSI, because we've not said thought, stress, and immunity all out loud for probably 20 years. It is an older study and started in 2001 with funding from the Dana Foundation. And then it was funded through 2023, I think, 2024, with funding from the National Institute on Aging. At one time or another, we've had over 300 healthy-ish, community-dwelling older adults in the study, not all at the same time. When you study aging, you have to refresh your sample. People get ill, they die. But overall, at all points of the study, we've had over 300. Their average age is 75, but they are across a wide range of ages in older adulthood, so from 60 all the way up through 93 at study entry. They are, like most research volunteers, generally well educated, and they're also mostly white, and they're about 57% female, which is true of the proportion of men and women in older adulthood. They underwent semi-annual assessments. So an interviewer went out to their house, administered scales, gave neuropsychological testing, and then after that assessment, a nurse went out and drew their blood. So we have measures of their immunological functioning as well. So I'm going to start with a study of learning and memory and the effects of cortisol, the hormone cortisol. Now, recall that inflammation is associated with risk for Alzheimer's disease. And one mechanism for that might be this hormone cortisol, which is anti-inflammatory. When there is chronic inflammation, however, cortisol rises with the inflammation, but it is ultimately unsuccessful at containing that inflammation. But it may have adverse effects because cortisol is associated with neuronal loss in the brain, particularly in regions associated with learning and memory, such as the hippocampus and the prefrontal cortex. So this analysis used 132 people from TSI. It encompassed 419 annual episodic memory testings between 2006 and 2011. They were administered the Ray Auditory Verbal Learning Test, which is a wordless learning task. You're given a list of 15 words, and you have to repeat them back. You're given the list 5 times. The number of words you recall across those 5 trials is total learning. You then go away and do other stuff for 20 minutes. You're asked to remember as many words as you can. That is delayed recall. And then they also collected saliva samples at waking 5 p.m. and 9 p.m. on 3 consecutive days. And we assayed those samples for cortisol and created an area under the curve. Now, cortisol has what we call physiometrics. It has measurement properties. There's a whole other soapbox I could get on about the measurement properties of various kinds of biomarkers, but you don't want that because I get real passionate and real preachy about it. It's not very much fun. But cortisol was reliable across all of the measurements to tell the difference between me and you, between person individual differences. The reliability was lower, but sort of okay for within person change. That is the difference between me now and me at a different time point. So a lot of the studies that I'm going to be showing you are actually multi-level models where we're separating out these individual differences and changes over time. All right, let's get to this. So these are the results. These between-person individual differences in cortisol were associated with poorer learning, that is, getting things into memory. But what's interesting is that the effect differed based on word position. So you probably remember the primacy and recency effect, like from Introduction to Psychology. And here, what we see is that cortisol has no effect on the most recent words. So when I read you this list, the first words you're going to say back to me are actually the last words on the list because they're still in your working memory. So you're just going to spit those out before those get lost. And then you're going to start telling me about words in the earlier part of the list. Those words have to get into longer term memory in order for you to recall them even seconds or minutes later. And it's these words, the ones that had to be put in long-term memory, that were associated with having higher cortisol. And what's more, this difference, where you still have good recency effects, but earlier long-term memory effects are impaired, is characteristic of pathological memory conditions such as Alzheimer's disease. Okay, so a few years ago, NIA commissioned a report on the relationship between personality and Alzheimer's disease. And, you know, I was known for working personality and health, and they asked me to do it. I wasn't explicitly studying Alzheimer's disease, but I thought like this was an interesting question. And so I took the gig. And I borrowed from the literature on personality and psychopathology to use 4 models to think about the relationship between personality and Alzheimer's disease. So one of those is the common cause model, where personality and Alzheimer's disease have a common cause but are not causally related to each other. For example, you have a high-risk gene for Alzheimer's disease that is affecting that, but maybe also influences your personality. And so it looks like personality and Alzheimer's disease are related, but it's really an epiphenomenon of this genotype that you have. There's not really any evidence for that kind of model. The predisposition model says that personality affects risk for Alzheimer's through some plausible pathway. And an example would be personality affects health behaviors that modify risk for Alzheimer's disease. And there's lots of good evidence for this. I'll show you some of that evidence in a minute. Most of the evidence is for the personality dimensions of neuroticism and conscientiousness. Things like agreeableness, extroversion, openness, intellect, those are less strongly and kind of inconsistently associated, but there are very robust relationships for neuroticism and conscientiousness. The model that really, really interests me is called the pathoplastic model. And it says that personality affects the appearance or severity of Alzheimer's disease. So, as an example, the cognitive or behavioral consequence of a particular level of pathological burden is determined by personality. So pathological burden could be the amount of Alzheimer's amyloid plaques in your brain, or the amount of tau tangles in your brain, or the presence of a high-risk gene. That the relationship between that risk and your clinical outcome depends on personality. So I'm going to talk about that more. And then there's the spectrum model that says personality is part of the spectrum of AD symptoms, and personality differences associated could be just prodromal symptoms of Alzheimer's disease. There is personality change in people who have Alzheimer's. Maybe it just looks like personality affects Alzheimer's because of these early personality changes, but there's not any good evidence for that because some of these relationships have been observed even when personality is measured decades before the clinical onset of Alzheimer's disease. So that seems to be unlikely. Most of the evidence is consistent with predisposition, some intriguing evidence for pathoplastic. What does a pathoplastic relationship look like? Here we have age on the x-axis, and we have the odds of Alzheimer's disease on the y-axis. These 2 lines on the bottom have 1 high-risk gene. People on the top have 2 high-risk genes. What this shows is a very well-known pathoplastic effect of sex. So you'll notice that in both for both genotypes, and at almost every age, women, given the same risk, are more likely to get Alzheimer's disease than men. That's a pathoplastic relationship. Sex is affecting the appearance of Alzheimer's disease given the same risk. And here's an example from personality. So these flat lines are people who do not have amyloid plaques in their brain. These 2 bottom lines are people who do have amyloid plaques. But you'll notice that people who are not anxious people have much less cognitive decline than people who are anxious people. So again, here personality is changing the process associated with a risk factor. The outcome is different, even though the risk factor is the same, and that's associated with personality. So let's talk about inflammation in the brain. If we're just thinking about predisposition, there are lots of meta-analyses and data syntheses showing that higher levels of inflammation are associated with higher risk of Alzheimer's disease, as I've been saying all along. Where they have looked at multiple measures of inflammation, the one that is most associated with risk is this one called interleukin-6 or IL-6. IL-6 is a pro-inflammatory cytokine. It's produced by immune cells as well as by fat cells. And it's so closely related to risk for age-related disease that it's been called a cytokine for gerontologists. So that's IL-6. And it can have pathoplastic effects as well. Or so this is the effect of IL-6 on the amount of growth of amyloid plaques in the brain. If you don't already have amyloid plaques, inflammation made no difference. If you did already have amyloid plaques, interleukin-6 greatly accelerated further deposition of plaques in the brain. So what I was interested in is there a pathoplastic relationship between IL-6 and cognitive decline, specifically memory decline, that is pathoplastic with personality? So this analysis used 256 people from TSI, 973 administrations of the RADLT between 2006 and 2023. Everybody filled out the NEO-FFI at baseline when they entered the study. And this analysis focused on neuroticism and conscientiousness. So neuroticism items are things like, I'm not a worrier, reversed. Sometimes I feel completely worthless. And conscientiousness is things like, I keep my belongings clean and neat, and I'm not a very methodical person, which would also be reversed. We had interleukin-6 both between and within people, the same way we did with cortisol. And we also had their expected memory performance based on regression norms that we put in the model. And so what we're trying to predict is, how are you doing relative to expectations? Was your memory function better than we would expect based on your age, gender, and IQ? Or was it worse than we would expect at that time point? Okay, and here's what we find. Not neuroticism, but conscientiousness did moderate effects of IL-6 on total learning, as well as on delayed recall, although the delayed recall fact is attenuated if you put total learning in the model, right? Because you can't recall words that you didn't learn. So part of the effect is due to the effect on total learning. In any case, this is showing you total learning. There was this modification, but it was not a pathoplastic modification. So the people at risk who had high average IL-6 pretty much tracked expected performance over time. It was for the people who had lower IL-6 that conscientiousness made a difference. So people who were low in conscientiousness also tracked expected performance over time, but people who had low IL-6 and were high in conscientiousness, expectations over time, let's just say with age, decline, but these people did not decline. They did better and better over time relative to these declining expectations. So it's not like their memory got like so much better. It's actually that they are maintaining more relative to these declining expectations. So not pathoplastic, but still interesting. And it was actually sort of a doubly pathoplastic way, pathoplastic in quotes because it's not really pathoplastic, but these are men on the top and women on the bottom. And even though this wasn't a per se pathoplastic relationship, women were still more sensitive to these effects than men. So that was quite interesting. All right, now I'm going to bring infection back into the picture. We know that infection is associated with immune aging and inflammation. Inflammation can affect the brain. So this is an analysis done with Justin Carr, who's a neuropsychologist at Kentucky, and Rebecca Reed, who was my postdoc and is now at University of Pittsburgh in the psychology department. And the study asked, can infection influence executive function and self-regulation? And we know that in young and midlife people, that latent infections, and these are all different kinds of latent infections, are associated with poor cognitive function. In this study, we focused on these two, cytomegalovirus, which you already know about, and which you already know can infect the brain directly, and a parasite called Toxoplasma gondii. Let me tell you a little bit about toxo. So it is an intracellular parasite. It infects the brain directly, and so this is a toxoplasma cyst in the brain of an immunosuppressed person and a lesion in the eye of an immunosuppressed person. But like CMV, if you're not immunosuppressed, you probably don't even know if you got it. Also, like CMV, once you are infected, you are always infected, and so the prevalence increases. The difference being that it doesn't increase as much. So only about 25% of people are infected by older age. The definitive host for toxoplasma is not humans, it's actually cats. Cats are the definitive host for toxoplasma. But the interest in the effects on the brain do not come from the cats. They actually come from mice. So mice, like humans, often get toxoplasma from contact with cat feces or infected soil, but it can also be sexually transmitted, by the way, also in humans, FYI. And then something happens when the mouse gets infected with toxoplasma. It loses its aversion to the smell of cats. And I learned this morning from Kieran that mice may actually even be attracted to the smell of cat urine. Now, this does not bode well for a mouse, right? They are more likely to be predated upon by a cat. The toxo gets back into a cat. And in this way, it perpetuates its own cycle of infection. And this has led to questions about whether there might be any kind of analogous effect in humans. Do humans become less fearful, more impulsive, and so on? And there's even like a book that says, yeah, we think there is. Okay. So this is 138 people in this analysis. We knew their CMV and toxoplasma serostatus. We measured their executive function. We have 1148 observations of executive function, and self-reported problems in self-regulation was given annually, so we have 645 observations of self-regulation. Seroprevalence in the sample, very much like what we would expect in a population of older adults. Most people infected with CMV, most people not infected with toxo, and these were unrelated to each other and unrelated to sex or gender. Okay, our measure of executive function was from 4 neuropsychological tests that measured things like cognitive flexibility, working memory, and mental manipulation. And this composite was reliable both between and to a lesser degree within people. And here are the effects of latent infection on executive function. No, that's right. There were none. We did see that this composite was lower for people who were older, and it also declined over time. So the composite was sensitive, it just wasn't sensitive to infection. We also had this measure of self-regulation, which was the behavior rating inventory of executive function, but it's not really executive function. What it really is problems with impulses and emotions in the behavioral regulation domain. I rush through things, I have emotional outbursts, I talk at the wrong time, and problems keeping on track with activities and tasks in the metacognition domain. I have trouble getting started, I have trouble prioritizing or finding things. And this scale was reliable both between and within people. This graph shows data from people who have CMV in red and people who do not have CMV in black over time. So this is up to 15 years. This particular shows metacognition, similar effects for behavior regulation. People who had CMV reported more problems with self-regulation than people who did not have CMV. And there was some indication that as time went on, people with CMV had more and more problems, while people without CMV remained mostly stable. Here are the effects of toxoplasma on self-regulation. For women, if they had toxoplasma, they reported more problems with self-regulation. For men, if they had toxoplasma, they reported fewer. This is opposite of what the canonical effect of toxoplasma is. The canonical effect is that men are more vulnerable, and when they are infected, they have more problems with self-regulation. They're more impulsive, they're more uncontrolled, not less. And this brings up the issue of the difference between younger and older people. So we don't know the exact mechanism of how toxoplasma works in the brain, but it's thought to have effects on testosterone and dopamine in the brain. And it also harvests folate from neural cells, which is not good for them in the least. So if we think about men, for younger men, if there's some influence on testosterone that makes them more impulsive, that would be an effect. For older men, though, maybe more testosterone helps them. Maybe they are less fatigued and less likely to make errors in self-regulation just because they're tired. So their baseline level of testosterone is different from a younger person. And so this canonical story about the effects of toxoplasma on behavior maybe doesn't apply to older people. Maybe it only applies to the people who have been studied before, which are young men and young women. Why are women adversely affected? Women typically are not as affected as men. So we're thinking about like, why would they be affected in this case? I'm going to engage in some rank speculation. So don't quote me on this. I know it's being recorded, so that's kind of a problem. But one of the things I said that toxoplasma does is it harvests folate from neural cells. And older women might be vulnerable to this, in part because a good source of folate is enriched bread. And over time, the proportion of, they're called PWAGs here, these are people without celiac disease avoiding gluten. Even from 2009 to 2014, this number of people increased quite dramatically. And they are much more likely to be women than men. So if we have older women who are, for whatever reason, avoiding gluten and not getting enough folate, and now toxoplasma comes in and takes away more of their folate in the brain, you know, maybe that's a reason why. That's rank speculation at this point. So in the last phase of the TSI study, we had a subset of people who had neuroimaging. And I actually meant for this to be the last slide. I was going to tell you about, my collaborators. This is Brian Gold at University of Kentucky, John Haken at Penn State, and Pete Giannaros at Pitt. Because I'm not a neuroimager, you have to work with the experts. So this was going to be like coming attractions, right? But just last week, literally last week, my Oregon State student, Ava Barnes, and I started analyzing some of the neuroimaging data and looking at the relationship between conscientiousness and neuroimaging. The subsample had 80 people in it. We have data from 180 structural MRIs. So people had MRI done every other year, up to 4 times. And we had, again, conscientiousness from the NEO. Reminder, I keep my belongings clean and neat, being methodical. And then we had regional volumes from structural MRI that were in regions where loss of volume has been associated with risk for Alzheimer's disease and dementia. So we're looking at like target mediators that could lead to the development of dementia. And this is what we found. So this is showing you change in the hippocampus over time. The top line are people high in conscientiousness. The bottom people are people low in conscientiousness. And you can see that people low in conscientiousness are losing hippocampal volume faster than the people high in conscientiousness. And what we're really interested in is this change over time, because most of the studies have been cross-sectional and they've been inconsistent in terms of what regions have been associated with personality. But in this case, we see this effect for hippocampal volume. We see this in the entorhinal cortex, which is a structure very close to the hippocampus and as sort of a pathway for information going to the hippocampus. We see it in the general temporal cortex, but we do not see it in the frontal cortex. We see individual differences in the frontal cortex, but we don't see differences in rate of change. And this is really the thing that's problematic, is when you're losing volume. And so we're very excited about this. This is one of those findings where we saw this. I was flabbergasted. I put the slide in. I slept on it. I got up in the morning and controlled for demographics. It's remained. I slept on it again. I got up in the morning. I did a bunch of scatter plots looking for any kind of outliers. There weren't any. And so I have some good confidence in this finding. So now that's supposed to be the end. But I have a little bit of time. And so I'm going to give you a little bit of a UBC special Easter egg. Okay, when I was on internship here, I was thinking about this question that I had been posed. So I've been doing some work with worry. And somebody said to me, well, what's the difference between worry and rumination? And I was like, well, worry is future focus, blah, blah, blah. And rumination is this. And they were like, yeah, but what's the correlation? And I was like, I don't know. I don't know what the correlation is, but I'm going to find out. I went and looked in the literature. I couldn't find one. I couldn't find a correlation between worry and rumination. And so when I got here, Lynn Alden was my research supervisor, and we designed this study where we looked at worry and rumination and negative mood. And what we found is that it was generally the shared variance that was associated with negative mood. It wasn't anything specific to worry. It wasn't anything specific to rumination. It was a nice SEM model, and it all went through the shared variance to both depression and anxiety. So the idea that worry is the thing that causes anxiety and rumination is the thing that causes depression, not at all evident in this. I expanded the model to more kinds of repetitive thought, not just negative kinds, but now also positive kinds, things like reflection and reminiscing and emotional processing And along with a couple of other people, Lynn and I wrote this paper on a multidimensional structure for repetitive thought. So this is the structure. This is in older adults. It replicates the structural finding that we had in younger adults. And it's basically a three-dimensional model. You only see two here. I'll tell you why. One is positive to negative. So you see you're doing like positive kinds of repetitive thought over here, and you're doing negative kinds of repetitive thought over here. This dimension we call searching solving. Searching things are like uncertain or questioning or exploring. Solving are things that are sort of more certain or declarative as knowledge. And then it turns out that some people do it all. They are just real thinky people. And then there are other people who are not real thinky people. Like they just don't engage in very much of this at all. And so we have this dimension that's called total, which captures this individual difference in like how much you do positive and negative, every kind of repetitive thought versus not. Okay. We're working on a meta-analysis now and gave a whole slew, like every repetitive thought measure we could find. I've like flipped this and inverted this so that the dimensions are the same as I showed you before. If you're thinking about developing a measure of negative repetitive thought, please stop and do something else. We have so many measures of negative repetitive thought, and they're all close to some other measure that already exists. So we don't need any more. But what we need, we could use some more measures of like reappraisal, distraction, reflection, problem solving, all that kind of stuff. We don't have very many measures of that. But in any case, we still get the same structure. So what do we know about repetitive thought from memes? It's called in memes overthinking, and it's bad for you. So you've got to stop overthinking. You're going to break your own heart. I don't know where this statistic comes from, but apparently it's the main cause of people being stressed. So I'm just going to show you one psychoneuroimmunology study using this repetitive thought framework. And this was motivated by the relationship between IQ and diseases that are also associated with inflammation. So people who are higher in IQ have lower risk for things like heart disease, for example, infectious disease, for example. And so we sort of, I'm flipping the arrow now, where I've been talking about immune effects on the brain. Now I'm going to talk about IQ effects on inflammation. And whether that is moderated by or mediated by repetitive thought. So this analysis had 120 people. We had 804 biannual measurements of interleukin-6. We had estimated full-scale IQ from the North American Adult Reading Test that they took at study entry. And then we also had dimension scores from that multidimensional scaling that I showed you. So you take each of those individual points, weight it by the dimension, where it is on the dimension, right? So something way at the end would be highly weighted. Something in the middle would not be very much weighted on that dimension. And you get a dimension score. So we had trait repetitive thought dimensions from the multidimensional scaling. And what we found is that, in fact, well, let me first say, in this sample, I have high IQ and low IQ, which is actually average IQ, right? So this well-educated volunteer sample, you have some very smart people and some sort of regular smart people. But the very smart people had lower inflammation than the regular smart people, unless the regular smart people were also engaging a lot of total repetitive thought. And we could say here that because there's actually a relationship between IQ and total, like people who are higher in IQ tend to endorse more total repetitive thought, that if you take someone who's average IQ, but they're thinking like someone who's high in IQ, that you see that they also have the same level of inflammation as someone who is high in IQ. So that's your little Easter egg. So this is actually what the memes should be saying. So you can't see this very well, but it says, here's what all intelligent people do: talking to yourself, defining your problems. And this is one of my favorite tweets from Ginny Hogan. Not overthinking is hard for me. I'm really good at thinking. And this very much captures this relationship between the amount of repetitive thought that people have and their cognitive abilities. All right, so this actually really is the end. So I want to thank my collaborators. I want to thank my labs at UK who worked on the thought, stress, and immunity study. Ava, who has started working on the brain data with me. I thank the Dana Foundation and NIA for funding all of this work. And I thank you for your attention. Thank you very much.
Okay, so thank you so much for your amazing talk. Now we have time, lots of time for Q&A, and we are going to use this so that the recording will have the questions. So we have a few of these around that we can also pass. So if you get past one and it's not on, there should be a little green button. Just hold the button, turn it on. But go for it. Any questions?
Yes, please. Wait, yeah, with your mic, I guess.
First off, thank you so much. That was a wonderful talk and so engaging. It felt like it was 5 minutes and it's over. Lots of questions, but I guess a practical takeaway that other people might be wondering too is like, is there a vaccine for this virus that everyone gets?
Yeah, I always get this question. That's a great question. The question is, isn't there a vaccine for cytomegalovirus? And the answer is no, there is not, which kind of surprises me because not only is it driving immunological aging, making older adults more vulnerable, but it's also, after like genetic reasons, it's the #1 leading cause of deafness in neonates. So there's another reason why you might want to be vaccinated against CMV, but there is not a vaccine against it. That's surprising. Maybe because people don't think about it. Like, it's kind of invisible, right? You get it, you don't know that you have it. It's aging your immune system, you don't know why. Like, it's not like something that, you know, really hits you like COVID, for example. But it would be great if there were, because I think we could keep older adults healthier for longer, potentially, if we could. We would have to vaccinate very early, right? Because you remember, like, even about 1/3 of little kids have CMV. So you'd have to vaccinate quite early to be really preventative. But there's no reason why that couldn't be done like you vaccinate for other childhood infections. By the way, probably if you didn't get CMV in childhood, you got it from a child, because they are little virus bombs, right? When they get CMV, they shed way more virus than an adult does. And they also stay infectious longer than an adult does. And CMV is transmitted through body fluids, which children are happy to share with you on any occasion. And so you see situations where, like, a parent will get it from a child who was at daycare and was like, you know, kids are touching each other's faces and sharing fluids in various ways. And then the kid gets CMV, and then they're infectious, but nobody knows it. And then they come home, and if the parents haven't already been infected, the parents get infected. And there's also an SES gradient. So almost everybody gets CMV, 80% of people by the time they're old. People who have lower SES tend to get it earlier than people who are of higher SES. So, CMV, it's not great.
I was super interested in the finding on like the hippocampal volume and the con. I was wondering what the measure was for like when you're doing conscientiousness, because I was how like having low hippocampal volume could lead to less conscientiousness because you organized or so. I know you said they're not one-way arrows, but what's
Yeah, it's interesting because when we were designing this study, there were sort of two different things about the brain that we wanted to look at. We wanted to look at some brain stuff that might support conscientiousness and self-regulation, which would be more frontal regions and more connectivity. And then we had hypotheses about the brain that had to do with not disease progression per se, but sort of pathological changes like high volumetric loss, which were different regions, right? So we were thinking more about like frontal regions and connectivity for things that would support conscientiousness and self-regulation. And then when we thought about the effects of conscientiousness and self-regulation, we were thinking more like midbrain temporal kinds of areas. So in fact, the finding that this relationship was related to change in those areas, but not in the frontal area, is in line with our conceptualization of it. Does that mean that it's not possible? It's an observational study. Anything's possible. But the way that we had it conceptualized is consistent with this, with conscientiousness having more of a causal role. Now, recall that I said that there were individual differences in frontal volumes with conscientiousness, but not change over time. So that would be one where you would, I think, very genuinely ask, yeah, is that a conscientiousness effect? Or is that somebody with, and it's all adjusting for intracranial volume and all of this other stuff, but just having like a big brain up here help you be more conscientious, and that could be very much a yes answer. Yeah, thank you.
Hi, very interesting, thank you. I have a question about your last slide. It's really interesting to see that like thinking more frequently is associated with less inflammation, but I'm wondering that does include both positive and negative aspects, right? So like catastrophizing, rumination, all of that. I'm curious if you looked at it broken down by like positive or negative.
Yeah, so we looked at all three dimensions. We looked at the valence dimension, it didn't do anything. We looked at the purpose dimension, it didn't do anything. We're still trying to figure out exactly what the purpose dimension does, because it doesn't correlate with a whole lot of stuff. But it was only the total dimension that did this. So there might be some, and in terms of correlates of total, we found IQ and we found neuroticism. So do we know that the exact content of even negative thoughts is the same for someone who's high in neuroticism and someone who has high IQ? We do not know that. We can't like drill down into their brains, or we haven't yet, to see like what are your exact thoughts. Like when you're thinking about worry, maybe there's something very subtle about the worry of someone who's high in IQ that is more productive than the worry of someone who's high in neuroticism. But yeah, that's a great question. We did look at that. Yeah.
Thank you for the talk. It was very interesting. I was like, so you're talking about like personality and like these different potential pathways that might be, I guess, related to Alzheimer's. I was interested in, I think one thing that was kind of surprising to me was like openness not being part of that story, especially, I mean, given you kind of have these later findings of like IQ and inflammation. My understanding is like IQ and openness are tend to be correlated. And I also, when I think of potential protective factors for Alzheimer's, at least one thing I've heard, and you probably know more than me, but like the idea of like being like a lifelong learner, someone who is engaged in learning. So I was just wondering if there haven't been like effects related to openness in the field, or like could you maybe talk about that?
In the meta-analyses and in the larger studies, neuroticism and conscientiousness overwhelm everything else, which is surprising because we know like IQ is a protective factor. But let's think about this for a minute. I showed you that picture of Justin Carr, the neuropsychologist that I worked with. One of the things that he's interested in is measuring cognitive decline in high functioning people. So if you think about a sort of standard neuropsychological approach, a standard neuropsychological approach is we have a threshold, like here's the average, and it's usually like 1.5 standard deviations below the average. And once you drop below there, then you're considered to have some kind of memory problem, for example, or an executive function problem, whatever it is. But some people start here and other people start up here. And so they will have a lot more decline before they get to that level than people who start here. So 2 people might start, and this person is going to get to the pathological level before this person does. But they're both declining at the same rate. So this is kind of an intercept question. And so is IQ associated with risk for Alzheimer's disease because you're actually just starting at a higher level than people who have average IQ even. So he actually has this method for testing cognitive decline based on the number of high scores. And he has norms for if you give 7 tests and you have a college degree and an IQ of 115, how many of those tests are you expected to have a higher than 90 percentile score for? And so if you're that person and all of your scores are like 50 to 60th percentile, even though in a neuropsychological assessment, we would say you're perfectly normal, from this framework, we would say this person has probably experienced considerable cognitive decline, and we should be paying attention. So this is a long answer to an easy question, which is there this relationship? And there's generally not. But I think there are these complications about when you're talking about like IQ and openness, intellect, Alzheimer's disease. I think this intercept question really becomes part of the problem that you have to account for. So maybe it takes you longer to get Alzheimer's disease, but ultimately your risk isn't influenced. Like the disease process, it doesn't influence the disease process. Can we say that? Yeah.
Thank you.
Thank you so much for that talk, Suzanne. You covered so much ground and so many exciting findings that I'm a bit curious about what you think is kind of the big picture for future directions. Also, I see so many of our graduate students and undergrads in this room, and like I'm curious what you think in terms of where the most bang for our buck as a field is, in terms of the effects you've been talking about. Like, and I guess to, where I'm sort of getting at this also more concretely is, maybe I'm thinking about a combination of where the biggest effect sizes are, plus like follow up on Liz's point about the fact that CMV is so common, like the base rates, right? So even if the effect size is smaller, the fact that so many older adults will have CMV, like maybe the greatest bang for your buck is there. And then finally, like another factor may be the malleability, right? Like some of these things that you've spoken about are more or less malleable, more or less influenceable by our lifestyle choices and so on.
Yeah. So I think if we take 2 models, we take the predisposition model, where you're talking about personality being a risk for cognitive decline in Alzheimer's disease through some plausible pathway. There are a number of those. There was a great paper that looked at the population attributable risk to modifiable factors. And those were things like physical activity, depression, cholesterol, I think, blood pressure. There was a whole slew of them, and a large amount of population attributable risk for Alzheimer's disease are these modifiable factors. Not most, but quite a lot. And so I think one future direction is trying to drill down and see what are these modifiable factors that are involved in these relationships. That's not to say that personality itself is not modifiable. There's actually quite a literature building up on intentional personality change. Personality naturally changes, both across the lifespan and actually moment to moment. So I don't know, has anybody ever read Will Fleeson's work? A couple of people have seen Fleeson's work. He does these great, or I don't know what he's doing today, but he has done these great studies where he took personality measures and modified them to be like in the past hour, right? In the past hour, let's take a look at some items. In the past hour, have you kept your belongings clean and neat? In the past hour, have you been methodical? And in fact, people, this is a reasonable thing. People can respond to these questions in a rational way. And what he gets out of that, that's so cool, is a distribution of personality. And if you look at people who score high, say, in extroversion on a trait measure, the mean of their distribution is higher than the people who scored more introvert on the trait measure. But the distributions almost entirely overlap. So someone who's introverted sometimes acts very extroverted. Someone who's extroverted acts sometimes very introverted. And so if you think about personality change, like intentional personality change, it's not like you're trying to roll a boulder uphill. It's that you're trying to shift that distribution. Introverted people act extroverted sometimes. If someone feels like, oh, it's a goal of mine to be more extroverted, all they have to do is, they're already doing that. They're already acting extroverted. They just need to do it more often. So there is this, as I said, this building literature on intentional personality change. I think in the personality and health literature, this would be a major contribution to show that intentional personality change has downstream effects for health. Because we have questions like this, like which came first, the structural brain differences or conscientiousness? If you can change conscientiousness and show effects, then we start to be able to make more robust causal inferences. So intentional personality change, you guys, would be huge. UB and JPS would just like that. Oh, just your last. I thought you had a question.
I can ask one.
Okay.
So speaking of the room of grad students and undergrads, which is amazing to see, your background and interests, very interdisciplinary, clinical, health, psychometrics, biostatistics. So how were you able to integrate all of those and have such a successful career? And even in your recent years in HDFS, so across your kind of career, how have you done so?
Not all at once. Let me just say that this is, it seems like, oh, you're doing neuroimaging and immunology and biostatistics and this and that and the other. I was not always doing all of those things. When I was in graduate school, I dabbled a little bit in statistics. I had that minor. I thought it was interesting. In fact, actually, nobody told me that you could do quantitative psychology or I might have done that instead of clinical. But in any case, I was doing clinical and I got some immunology training. I got a little bit of an immunology background in addition to my regular training, so I was able to do psychoneuroimmunology work. And, you know, that was it for a while. And then NIMH kind of changed its focus from including psychological health to only being interested in funding studies of mental illness. And so two things happened. One thing is I was trying to figure out who would fund my work because it was mostly on like optimism at that time. And I wanted to keep my optimism work going. But also I went to a talk actually by the immunologist whose slide I showed you, the massive proliferation slide. That is literally her slide. I went to a talk, she was giving a talk at UK, it was about immunology. I'm like, oh, this is interesting. Well, she's one of the leading experts on immunological aging. And she started talking about like viruses and immunosenescence and changes in the immune system. And I was like, that's fascinating. So I started to think about moving more into aging so that I could study that. And also then National Institute on Aging is more interested in like healthy aging and not just pathological aging. So that was another thing. So then I had PNI and then I had aging. And then I had like a K award. And while I was doing that, I was able to get my MPH with the biostatistics. So that was years later. And then we started doing this work. I started getting more interested in the cognitive stuff. And then I started collaborating. In fact, when John Haken was a postdoc, he actually came to me and said, hey, can we write a supplement to your grant to do some neuroimaging, and here's why. And so we did that. That got funded. We did a little neuroimaging, and then we made that a big part of the renewal for the next 5 years. So it wasn't like I went to graduate school and I did all of this stuff while I was in graduate school. I did like one thing when I was in graduate school, like 1.5 things, right? And then things have sort of added on as my career has progressed, as I've pursued different things and been interested in different things, and people have come to me with interesting questions that I've been interested in collaborating on, and I've had different opportunities. So that's how you do it. I mean, I've been a full professor for a while, and I've had time to accumulate those things. So don't feel like, oh, I have to do everything before I get my PhD. I can have that kind of career. Do like 1 or 2 things really well, and other things will find you as your career develops.
Excellent advice. Any last minute question? All right, I think we can give a big thank you.
Thank you. Thank you all.